The title of today's presentation is an overview of mycosis. The aim of this presentation is to enable students to be able to recall the basic ideas discussed during the physical classroom lectures. Now, the term mycosis refers to fungal diseases of man and animals. The ability of fungi to establish a disease depend on the following factors. Number one, virulence of the fungi. The virulence is the capability or possession of certain characteristics that enable a particular pathogen to cause diseases. Virulence of the fungi determine whether they can be able to establish diseases in healthy hosts or in immunocompromised patients. Another important factor is the dose. Dose is generally the minimum number of the organism required to establish a disease. So if the dosage is also called the infective dose, if the infective dose is not met, then disease cannot be established. Another important factor is that the fungi must be able to pass through the correct route of entry. For any pathogen to establish a disease, it has to pass through the right route of entry, either the gastrointestinal tract, genitourinary tract, respiratory tract. So each pathogen has specific routes in the body through which it has to pass in order to reach its target site where it can establish a disease. The last factor is the immunologic status of the host, whether the host is a healthy person with active immunity, whether the host is immunocompromised person, such as people at the extreme ages, neonates, old people, people that undergo chemotherapy, and so on. Now, let's look at the conditions that make a host or a patient or a person to be immunocompromised. There are several conditions that can make a person to become immunocompromised. Some of these conditions include cases of prolonged chemotherapy with broad-spectrum antibiotics, which serve to reduce or inhibit the growth of normal flora in the body, which provide protection to the particular site of the body. So when the normal flora population reduces, this normal flora generally serves to compete with pathogenic microorganisms at their site. So if the population is reduced due to the prolonged chemotherapy, then the person will become vulnerable or susceptible to being attacked by pathogenic microorganisms attacking those particular body sites. Other factors include cases of irradiation, use of immunosuppressive agents, use of broad-spectrum antibiotics, cases of cancer, cases of burns that burn the skin, and so on. organ transplantation in which immune suppression is required, mucosal disruption by any physical agents, metabolic diseases such as diabetes, which renders the person to become immunocompromised, cases of malnutrition in which the immune system is also affected, production of the white blood cells is reduced as a result of poor nutrition, immune deficiency syndrome caused by certain agents such as HIV virus that attacks the immune system, making the immune system deficient, abdominal or cardiac surgery, repeated intravenous injection, hematological dysfunction, and so on. All these are conditions that makes a person to become immunocompromised. And when such condition occur, many pathogens could have the potential to attack the body because the immune system has already been compromised. Now, let's look at the determinant factors of mycosis. Usually, category of fungal colonization and disease development usually depends on certain the body sites that is involved. So we can categorize fungal infection as either superficial, occurring on the surface of the skin, or deep tissue infection. However, these infections can generally be mild and self-limited. Most of the fungal infection are generally mild infection. The most affected parts include the superficial layer of the skin, and the disease are called superficial mycosis. Then the second part is the cutaneous layer or dermal layer of the skin. That is the layer beneath the superficial or upper epidermis, and the disease is called cutaneous mycosis. Then we have the subcutaneous, which affects the subcutaneous tissues beneath the skin. And then we also have those affecting the mucosal surfaces. And we have the deep tissue mycosis or systemic mycosis. Now, in order to understand mycosis very well, we need to have a recap on the skin anatomy itself, because most mycosis diseases are attached to skin sites, either upper layer of the skin or the middle layer or the subcutaneous layer of the skin. So skin is generally the largest organ that covers the subcutaneous tissues, and it is involved in temperature control, excretion, sensation, etc. So it carries out a lot of functions. It is generally made up of two layers, that is epidermis and dermis. Epidermis is the upper layer, and it is made up of 5 different sublayers, while the dermis is the layer below the epidermis. It is the main layer of the skin and is made up of 2 sublayers. Now, the epidermal layer consists of 5 sublayers. As I mentioned, we have the uppermost sublayer, which consists of dead cells and is called stratum corneum. We have the layer that follows it, which is stratum lucidum. We have stratum granulosum, with stratum spinosum, and stratum basale. All these are sublayers of the epidermis. And now let's look at the various fungal diseases based on their location on the skin. As we said, we have the superficial layer, which is the skin layer containing mostly dead cells. This layer does not have sensory cells. Uh, it doesn't have most of the sensory receptors attached to it, and therefore most of the infections that occur or limited to this particular layer do not attract immune response. Now, fungal infections affecting the uppermost layer of the skin or the superficial layer are called superficial mycosis, which are generally of three different types. We have the pityriasis or tinea versicolor, we have tinea nigra, we have phaedra, which can be white or black phaedra. All these we are going to see them in more details. We have the cutaneous infections which affect the cutaneous layer of the skin or the dermal layer of the skin. This layer contains living cells, and therefore these diseases attract immune response. They attract inflammation and other immune responses, and they are divided into three different categories. We have the infection of the skin, we have the infection of the hairs, we have the infection of the nails. All this we are going to see them in more details. Then we have infections affecting the subcutaneous tissues, which the most common ones are two, sporotrichosis and mycosis or chromoblastomycosis. Then we have the systemic infection or deep tissue infection affecting the internal organs of the body. Now, having seen the various locations of the fungal infection, let us have a quick look at the fungi responsible for this infection. Now, generally, fungi are divided into two: the multicellular fungi, which are called molds, and the unicellular fungi, which are called yeast. Now, the molds are divided into two based on their type of hyphae. We have those that have hyphae that is divided into in which the cells are divided by means of cross walls or septa. They are the mold known as they have what is called accepted hyphae, and we have group of molds that have we have group of they are called the septed hyphae mold, and we have those without the cross wall, which are called the accepted hyphae mold. Now, accepted hyphae mold generally cause two types of infections: the subcutaneous mycosis, as we can see the names of the species, and then the systemic mycosis. These are the species Rhizopus, Absidia, and Muco. Then we have molds that have septed hyphae are further subdivided into two. Those that are called monomorphic mold, which exhibit only the multicellular form. They retain in their multicellular form of mold, and they are responsible for superficial, they are involved in superficial mycosis and subcutaneous mycosis, as we can see. Then we have molds that are capable of existing in two different morphological forms. They can exist either in multicellular forms or fragment into unicellular forms. These molds are called dimorphic molds, and they are responsible for most of the systemic infection because of their of their ability, their ability to change, their dimorphic ability, their dimorphism, which gives them the ability to change into unicellular form so that they can spread to affect various organs of the body, causing a lot of systemic mycosis. They also cause superficial mycosis, but most of them cause systemic mycosis. Then in the case of the yeast fungi, we have those with hyphae-like projections, which is called pulse hyphae or pseudohyphae. These ones cause systemic mycosis. Cryptococcus neoformans is in this category. And then we have those the first one are those without pseudohyphae, which is the Cryptococcus neoformans, and then we have those that produce pseudohyphae, which are the yeast family, and they are responsible for cutaneous and systemic mycosis. So now let's look at the various mycotic diseases in more detail. Now, starting with the superficial mycosis. Superficial mycosis are generally mild but chronic infections that affect the upper epidermis or dead skin layer, which is called stratum corneum, as we have seen, and hairs. They are exclusively confined to the dead surface of the skin and its appendages. Hence, there is no inflammatory response from the host in the case of these diseases because no sensory areas of the skin are involved in the infection. Now, however, this type of infection sometimes may become extensive to affect the dermal layer of the skin. In the cases of immunocompromised patients, the fungi will have the ability to affect the dermal layer of the skin. But in healthy person, the fungi do not have the capability to infect the dermal layer. They are limited to the superficial layer of the skin. Now, the first category in this is the pityriasis versicolor, or sometimes called tinea versicolor. Now, this type of infection, as we said, is otherwise called tinea versicolor because the causative fungi grow radially on the epidermis of the skin, producing circular growths that affect melanin responsible for skin color, leading to the either hypo or hyperpigmentation of the affected area, and that will that pigmentation or circular pigmentation generally resembles a ringworm, that is mothworm, and that is why the disease is referred to as tinea, or derived from the scientific name of the ringworm. Generally, this infection occurs in the neck, in the back, in the chest, and the shoulders. Now, the lesions due to tinea versicolor range from pink to yellow-brown, and that is why it is called versicolor, that is multiple colored infection. It produces various colors on the skin. Generally, tinea versicolor or pityriasis versicolor is caused by Malassezia species, of which Malassezia purpura is the most common among them. This fungi can generally be seen even in the skin scraping as clusters of budding yeast cells mixed with hyphae. Now, Malassezia purpura sometimes causes seborrheic dermatitis and dandruff. This dermatitis is usually in immunocompromised patients. So if the patient is immunocompromised, the pathogen has the M. purpura can now colonize the dermal layer, leading to dermatitis. So diagnosis of this particular infection is generally by collecting the skin scrapings, after which the scraps from the skin are treated with 20% potassium hydroxide, which will digest the keratin to expose the fungal hyphae. Subsequently, the sample can be viewed under ×10 and then ×40 objective lengths of the microscope. What will be observed? Short hyphae resembling spaghetti with spores, making it look like spaghetti with meatballs. In the case of culture, generally, Malassezia purpura is cultured on subbrow dextrose agar (SDA). That is, but it has to be polluted with olive oil because the fungi is a lipophilic fungi. So for treatment, the treatment of Malassezia purpura is generally using the azoles such as ketoconazoles and so on. Now, the next type of superficial mycosis is Tinea nigra palmaris. Tinea nigra palmaris, as the name implies, nigra means black, palmaris, the palm. So this is another topical infection that is characterized by black macular lesions on the palm or soles of the legs and hand with little inflammation and scaling. And just like Tinea versicolor, Tinea nigra is also confined to the uppermost layer of the skin, which is the stratum corneum. This particular infection is generally caused by Exophiala warnecki, which is a fungi that was formerly known as Hote warnecki, which is a black pigmented fungi that is generally found in soil and other environmental surfaces. So diagnosis of Tinea nigra is also by collecting the scraps from the affected area, treating with 20% potassium hydroxide to digest the keratin and expose the fungal hyphae, and then viewed under ×10 and ×40 objective lens of the microscope. So when viewing branch septate hyphae and budding yeast cells with melaninized cell walls, that is black cell walls, will be seen. Culture is done just like that of Malassezia purpura in SDA polluted with olive oil treatment using azoles, just like Malassezia purpura. Now, the last type of superficial mycosis is called pyoidra. This type of fungal infection is otherwise called trichosporosis, which is named based on the common species that causes it, Trichosporan species. Now, this infection is generally the infection of the hair shaft. The previous infections we discussed, superficial infections, are all both affect the skin surfaces. Tinea versicolor is for skin, Tinea nigra is also for skin, while pyoidra is an infection of the hair, which is characterized by black or white nodules attached to the hair shaft, and the black one is caused by Pyoidra hoti, while the white pyoidra is caused by Trichosporon begeli. Trichosporon begeli is the most common, and that is why pyoidra was named after this particular fungi, trichosporosis. Now, the diagnosis of these diseases is generally by collecting the hair nodules and then treating them with 20% potassium hydroxide to digest the keratin to expose the fungal hyphae, and then viewed under microscope just like the previous superficial mycosis. When that is done, a branch septate hyphae with ascospores will be seen. Treatment. Now, let us look at the second mycotic diseases, which is caused by fungi. This diseases, which is called cutaneous mycosis, is an infection of the dermal layer of the skin, that is the layer beneath the epidermis, and hairs or nails. And it is generally caused by a group of monomorphic keratinophilic fungi which belong to the phylum Deuteromycetes. These fungi are commonly called dermatophytes because they affect the dermal layer of the skin. They are named dermatophytes. Infection can also, cutaneous infection can also be an infection of the mucous membrane, which is caused by Candida species, which are unicellular fungi. Now, the infection due to dermatophyte is called dermatophytosis, while the infection due to Candida species is called candidiasis. Now let's look at dermatophytosis. Dermatophytosis is clinically referred to as tinea infection. Just as we explained, tinea means ringworm-like infection because the fungi grow radially to produce circular lesions that resemble the ringworm in appearance. So dermatophytosis, as we said, is caused by dermatophytes which are a group of fungi that infect the keratinized layer of the skin. These fungi include 17 species of microsporum species, which infect the hairs and skin only. And then we have the second category, which are the Epidermophytum flocosum. This particular species infects skin and nails. And then the third species consists of 22 species of Trichophyton. These species generally infect all the three areas of cutaneous mycosis. They infect the skin, the hairs, and the nails. Now, dermatophytes these dermatophytes are generally classified into three groups depending on their sources in the environment, depending on their natural habitats. We have dermatophytes which are called anthropophilic dermatophytes. These are dermatophytes that are restricted to human hosts, where they produce mild or chronic infection. And we have dermatophytes which are called zoophilic dermatophytes. These species are generally found in animals, as the name implies, zoo. They are found in animals and they are transmitted from animals to human beings. Then we have the third category, which are the geophilic dermatophytes. Geophilic dermatophytes are a group of dermatophytes that are saprophyte in the environment. They originate from the soil, but occasionally they can be transmitted to humans and animals, where they cause inflammatory reaction, where they cause inflammatory reaction depending on the side of the body involved. Now, looking at the transmission and pathogenesis of dermatophytosis, dermatophytes, as we have seen, are generally transmitted through direct contact with the source. Direct contact with the source of the fungi. In the case of geophilic species, they are transmitted from soil to human beings. In the case of anthropophilic species, they are transmitted from one person to another person or animal. And then we have the zoophilic species, which are transmitted from animals to humans. So generally, the transmission can also be through direct or indirect contact with infected skin or hair, like in clothing, in hair combs, hair brushes, caps, furnitures, bed linens, towels, and even hotel rugs. Now, depending on the species, the dermatophyte can remain in the environment for a long period of time, up to about 15 months. Now, regardless of the source of infection, the spores of the fungi settles on the skin or hair or nail, and once they settles on the skin or hair or nails. They germinate to form a mass of branching hyphae, which now grow out radially to produce the circular lesions. During this process, the organism secretes various proteinases enzymes, such as acid proteinases, elastase proteinases, keratinases, and many other proteinases that degrade the keratin on the skin or hairs or nails, uh, after which the fungi can utilize the keratin as nutrient to support their growth. Now, during this process, the fungi also produce metabolic end products. These metabolic end products are generally those that trigger the inflammatory responses around the lesions. So the lesions typically appear with inflammatory, inflamed cycles around them because of the uh, uh, metabolic end product produced by the fungi. Now, let us look at the clinical manifestation of dermatophytosis. Generally, dermatophytosis are best described based on the site of the infection. We have the first category, which is infection of the skin. Infection of the skin is generally characterized by circular dry lesions with slightly raised red scaly margins surrounded by red itchy skin. It should be recalled that dermatophytosis affects the dermal layer of the skin and therefore it elicits immune response. So the lesions typically appear in any location but are particularly common in moist, sweaty skin folds, such as the tinea corporis. As we said, the dermatophytosis are called tinea infection. So we have under the skin infection, the first category is tinea corporis. Tinea corporis is the ringworm infection that occur on the body part. It is an erythematous scaling that occur on the body, various parts of the body, such as the neck, the chest region, and then the abdomen. Now we have the tinea cruris. Tinea cruris is a skin infection that occur in the groin area. It is sometimes called the jock itch. It is also erythematous and it produces scales just like the tinea corporis. Then, of course, we have tinea manum. Tinea manum is an infection, a skin infection that occur on the hand. It's generally a ringworm infection occurring on the hand. And the last category under the skin cutaneous mycosis is tinea pedis. This is generally an a ringworm that occur on the soles, of legs. It is sometimes called the athlete foots. Now, diagnosis of this cutaneous mycosis, cutaneous skin mycosis, is generally similar to that of superficial mycosis. Now let's look at the second mycosis, which is an infection of the nails. Infections of the nails, which is called tinea unguium. This infection is also called onychomycosis. It is a chronic infection of the nails and nail bed, which mostly occur in the two nails. The distal part gradually breaks and crumbles. The affected nail will now become thickened, deformed, friable, and then discolored. The main causative agent of these diseases are the Tinea rubrum and Trichophyton rubrum and Trichophyton interdigitale. These are the most common dermatophytes that causes onychomycosis. Now the next type of cutaneous mycosis is the infection of the hairs. Infection of the hairs can be either within the hair or outside the hairs, or a pebbles infection. So an infection within the hair is called endothrics mycosis. In this type, the sporulation of the fungi is within the hair shaft, replacing the intercapillary keratin and then leaving the cortex of the hair intact. Scaling is less pronounced in this type of infection because it affects the hairs. The affected hair, which is very fragile, will finally break at the surface, leaving behind a tiny black dot or spot. Infection of the hair can also be ectotric, as we said, which in this case, the spores are formed outside the hair shaft and then spreading around into the follicle to penetrate the cortex of the hair. Scaling is less pronounced as well because it is limited to the hair. The hair will break above the skin surface, just as in the case of endothrics infection. Then the last one is pavus. Pavus is a type of endothric infection that is caused by Trichophyton scoelleni. This particular species causes the endothric mycosis, in which the hair doesn't break, but it is lost completely and permanently due to degeneration of the hair follicle, leading to the formation of sharp, delimited bald spot on the affected area, either the head or the affected area. So if the adjacent skin is involved, it may produce crusty lesions and scars. Now, generally, infection of the hair is of two major types. The first one is called tinea barbae or tinea sarcosis, or sometimes referred to as Mycosis barbae. This is generally a type of tinea infection that affects the beards. And then we have the second one, which is called tinea capitis or ringworm of the scalp. This one is generally a ringworm infection that affects the scalp, which can be pubis, it can be ectotric, it can be endotric. So in both cases, we can have the three different manifestations. So the next type of fungal infection is the subcutaneous mycosis. Subcutaneous mycosis is a chronic fungal infection beneath the skin. That is to say, it involves deeper layers of skin, that is the dermis, subcutaneous tissue, muscles, and fascia, and open muscle tissues. It is generally caused by saprophytic fungi that are found in the soil and vegetations. The infection is transmitted by direct or indirect implantation of the spores or mycelia fragment from the environment through either a puncture or wound in the skin. Therefore, man is generally, we can say man is an accidental host that become infected following inoculation of the fungal spores, bear some forms of trauma. So in addition, this infection is localized as the fungi is less invasive, so it remains localized to form small masses or granules. Hence, this infection is most tentatively identified by the presence of a characteristic tissue reaction or granule. Now, let's look at the example of this mycosis. The first one is chromoblastomycosis. Chromoblastomycosis is a subcutaneous infection which is caused by dematicious fungi, Poinsettia pedrosae, and Cladosporium carioni. This infection, as mentioned, follows implantation through superficial injury of the skin, and it is generally characterized by nodular or verrucoid ulcerated or crusty skin lesions on the exposed areas of the skin, particularly on the lower extremities. This is an example of the infection. it is most common in the legs or arms. Now, the next type of cutaneous mycosis is Mycetoma. Mycetoma is otherwise called Madura. Is otherwise called Madura foot. Is generally a chronic tumorous infection of the subcutaneous tissues and bone, producing in which the sinus tracts open on the skin and granules or grain-like with plasma, grain-like granules with plasma oozing out. This infection is generally caused by actinomycetes, which are which have both bacterial and fungal characteristics, and some fungi, which generally produce granules composed of hyaline septate branching hyphae in the infected area. So this is an infection. This is an example of this particular infection. As the name implies, it generally affects the foot. The granules are found in purulent foci surrounded by fibrosis and mononuclear cell inflammatory response, as can be seen in this particular picture. The infection occurs after localized minor trauma on the skin. Generally, this particular infection is limited, but sometimes hematogenous spreading may occur. That is, it can spread to the lymphatic system. As said, the foot and legs are the common site of this particular infection. Now, the last common subcutaneous infection we need to talk about is the sporotrichosis. Sporotrichosis is a lymphocutaneous disease. That is, a disease affecting both the skin and the lymphoids. So skin lesions generally progress to cause lymphangitis and regional lymphadenopathy. It is generally a disease that is caused by dimorphic fungus known as the Sporotrix chancyi, which is common in tropical and subtropical regions of the world. This particular fungi is also a geophilic fungi, which is found from soil plant debris such as stones, timber, hay, and straw. So the entry of this particular pathogen is through abrasion. When there is abrasion or pricking by the thorn, then the fungi become implemented and then the disease starts. So the disease therefore occurs at the distal extremities, either in the hands or in the foot, producing solitary cutaneous ulcer or nodules with raised nodular erythematous lesions that vary in size from tiny lesions up to bigger lesions up to about 2.2 to 4 centimeter in diameter.